Morning everyone, today we will be looking at gene regulation with the lac operon as an example. Your learning objectives for today are to describe the structure and function of an operon and to explain how gene expression is regulated using the lac operon as an example. The first thing I would like you to think about is this. If each cell in your body has the same DNA, why are there so many different types of cells? And the answer, you already know. We've already covered how cells become specialized. So you may want to try to answer this question by stopping the video now and see if you can get at least 4 of the bullet points. Okay, so the answers are: chemicals cause some genes to become activated while others remain inactivated. mRNA is only transcribed from the active genes. That's then translated into proteins, and the proteins are what modify the cell permanently in some way. That results in the cell having a particular function, and these modifications result in cells becoming differentiated and specialized. We're going to look into this process in more detail today to see what sorts of chemicals could be causing genes to become activated and what can cause them to become inactivated. So, there are a few types of genes that we need to know about. There are the structural genes and the regulatory genes. A structural gene is a gene that codes for RNA or a protein product, something that can actually become a physical structure. Regulatory genes are genes that code for a regulatory factor, like a regulatory protein. And these genes and the proteins that they produce control whether the structural genes actually get expressed, whether they actually get synthesized from the mRNA. So if you imagine this structural gene here, it will not get transcribed unless the regulatory gene allows it to. And I'll show you what I mean in a minute. So we started the lesson by mentioning the word operon. Now the word operon is just a fancy word to say a cluster of structural genes that are transcribed together. And we can kind of see this here. We've got this promoter region, an operator region, and the genes. There are 3 genes here represented by the 3 different colors. It's really important to remember the order of this. So the POG, I just remember it as POG, promoter, operator, and then the actual genes. So the promoter region is where RNA polymerase, the enzyme involved in protein synthesis, actually binds to the DNA to start the process of transcription. Okay, so this needs to be free for RNA polymerase to bind. The operator region is where a regulatory protein, like a repressor, something that will stop transcription, can bind. And then finally we've got this cluster of structural genes that code for the actual proteins or the subunits of a protein. So, let's say we've got a RNA polymerase binding to the promoter region of the operon. If there is nothing in its way, it will be able to move along the whole length and will be able to transcribe the mRNA from this DNA. That's what happens if there is nothing blocking it. However, there are instances where there is something in the way blocking this RNA polymerase, so it cannot do its job and transcribe the mRNA. So, somewhere further upstream of the DNA, it does not necessarily have to be connected here, there will be some DNA that codes for a repressor protein. So this is the regulatory gene here, and it's not connected here just to represent that it could be somewhere further up the stream. It doesn't have to be immediately next to our operon. So that repressor gene will be transcribed to make mRNA, and that mRNA will be translated to make this repressor protein, this regulatory protein. Now, if you remember, the repressor protein binds to the operator. And you can see here that as it binds to the operator, it does cover up a little bit of the promoter region. And that means that RNA polymerase now cannot really fully bind. And so it cannot go through the promoter, operator, and gene region. And that means that genes will not be transcribed because there's this repressor protein bound to the operator, stopping RNA polymerase from going through. Now, there are instances where there's a certain molecule present around this, and this is called an inducer molecule. This inducer molecule, if it's present, it binds to the repressor protein, changing its shape, forcing it to disassociate from the operator region so that the RNA polymerase can go through and can actually transcribe the genes. Okay, so in any given cell in your body, any genes that are not needed will have some kind of repressor protein in the operator region, whereas the genes that need to be transcribed will not have this repressor protein, so that RNA polymerase can go through and transcribe. What I would like you to do is have a look at the following diagram, stop the video here and try to label up what all of these different numbers mean. So the 1, 2, all the way through to 8, and you've got the words on the side, you just need to match them up. Okay, so we've got two examples here. We've got what happens in the absence of lactose and what happens in the presence of lactose. So we can see here that we've got this line of DNA with three main regions. Now, number one is at the protein, RNA polymerase, the protein that wants to go through and transcribe mRNA. In its way, we've got a repressor protein, something that's stopping it from being able to go through. Now, we know that RNA polymerase binds to the promoter, so 3 must be a promoter, and the repressor protein binds to the operator, which is right next to it. But when the lactose is present, we can see that it can bind to the repressor protein and cause it to disassociate from the operator region. And that's what's happened here, which means that lactose is an inducer molecule. And then after that, we've just got the gene 1, 2, and 3, the 3 structural genes. What I would like you to do is attempt this 4-marker question to check that you can now do learning objective 1. Describe the structure and function of an operon. Okay, and the answer is, an operon is a cluster of structural genes. Those genes are transcribed together. An operon contains a promoter and an operator region. You need both of these to get this mark. RNA polymerase binds to the promoter region. A repressor protein binds to the operator region. This regulates gene expression. And then you can give an example. If there is no repressor protein, then the gene is transcribed. If there is a repressor protein, then the gene will not be transcribed. Both of these are fine, but only one gets you a mark. So we're going to look into the lac operon as an example. This is an operon in E. coli, in the bacteria E. coli. And what it does is the lac operon contains 3 genes. It's lac Z, lac Y, and then there's also lac A. These genes, when they're transcribed, they make the following 2 proteins. This is beta-galactosidase and lactose permease. The beta-galactosidase catalyzes the hydrolysis of this glycosidic bond in lactose to make the monosaccharides galactose and glucose, whereas the lactose permease is a protein found in the membrane of E. coli, which helps them absorb or helps to transport the galactose and the glucose inside of the cell, so that the bacterial cells can use this for energy. Okay, so essentially, if there is lactose present, the bacteria will want to have these 2 genes transcribed so that it can actually make use of the lactose, break it down, hydrolyze it, and transport it into the cell. The first thing I'd like you to do, though, is do the following. Match the components with their functions, and then tell me what the functions of these 3 are. So if you stop the video here. Okay, so a structural gene is the gene that codes for actual lac enzymes in the lac operon example. Regulatory gene is one that produces a repressor protein. Then we've got a promoter. This is where RNA polymerase binds, and an operator where the repressor protein binds. The function of the repressor protein is to prevent RNA polymerase from transcribing the operon, or the cluster of structural genes. These 2 words are almost interchangeable. RNA polymerase is a protein that transcribes the DNA into a sequence of mRNA, and lactose binds to the repressor protein, causing the repressor to fold and become inactive. It's an inducer molecule. And we can see this diagram again, this time seeing it as the lac operon instead. So in this example, we would have the gene 1, 2, 3, but it's the lac Z, lac Y, and lac A. The lac Z and lac Y are the only ones you really need to worry about. Now to check that you remember the structure of the lac operon, I would like you to draw it here. It's worth 4 marks. So stop the video. Okay, and these are where the marks would be allocated. If you have drawn a repressor, so just a rectangular box with the word repressor in it, you get a mark, and it should be disconnected from the rest of your operon because it's somewhere further upstream in DNA. You get a mark if you've got the the promoter. You get a mark if you have the operator, and these must be in the correct order: promoter first, then operator, and then you need to have the lag Z, lag Y, lag A. All 3 of those are needed to get your mark here. However, you only need to know what the lag Z and lag Y make, which is the beta-galactosidase and lactose permease. So, let's have a look at what actually happens with the lac operon. So if the bacteria is found in an area where lactose is present, and the bacterium obviously wants to make use of this lactose, what we have is the inducer molecule, the lactose, binding to the repressor protein. Okay, that means that the repressor protein changes shape and so disassociates from the operator region, so it just leaves. The promoter is now unblocked because there isn't this repressor protein kind of covering it up. That means that RNA polymerase can bind to the promoter region, and it can now move through the operon and transcribe all of the structure genes, the lag Z, lag Y, and lag A. That means that the beta-galactosidase and lactose permease can now be synthesized. If there is no lactose, what we have is the regulatory gene that's further upstream is expressed and produces this repressor protein that sits on this operator and partly covers the promoter region as well. And that means that RNA polymerase can't bind, and if it can bind, it can't go through anyway. So the structural genes cannot be transcribed. They should say transcribed, not translated. Transcribed first, then translated, and so we say that they are switched off. Those are not active genes in that particular cell. Finally, I would like you to attempt this exam question. The control of the expression of the lac operon genes, which allow uptake and digestion of lactose in the bacterium E. coli, is well known. Figure 1.2 shows the arrangement of the elements of the lac operon. Describe how genes Z and Y are switched on in bacteria that are moved to a nutrient that contains lactose. Okay, so they were in an area where there was no lactose, but suddenly they have been moved to an area where there is lactose. So you need to tell me, in terms of the lac operon, what is happening to make sure that these genes are actually switched on. I would like you to write this on a Word document, or you can do it by hand and take a photo, and there will be a submission link for your work on Moodle. I would like you to submit that by 4:30 today, please. Thank you.